Good afternoon, everybody. Welcome to the HIRM RNA Technology Showcase. This is the first one for 2021, and we'll have a few additional RNA technology showcases through the year. This is an opportunity for pharma and biotech who have interesting technology that they want to share with us that could really help in RNA-based research. Give them an opportunity to come and tell us about some of this new technology. We like to hear how they're using the technology to advance RNA research or clinical research. This is an opportunity for us to ask some questions about it as well. So each talk will be about 25 minutes with 5 minutes for questions. And please feel free to sort of ask your questions either into the chat or unmute yourself at the end of the talk. So we're going to go in a slightly reversed order to what was initially shown. We're going to first hear from Dr. Emily Martersteck from Bio-Techne. She's a field application scientist, and she's going to talk to us about enabling spatial mapping and quantification of RNA in situ with RNAscope, miRNAscope, and BaseScope. Welcome, Emily.
Thank you so much. Let me see if I can share my screen. I hope that you can see this full screen now. Please let me know if not.
It looks good.
Okay, great. So actually no doctor in front of my name, but I am a field application scientist in the Boston area. And I am going to turn off my camera just for the presentation because I tend to have bandwidth issues if I try and run both at the same time. But then at the end to take questions, I can try and turn that back on. So let's see how that goes. The secret is out as far as spatial transcriptomics and how cool and how useful it is. It was actually Nature's editorial board's method of the year for last year. And it's such a powerful tool because not only do you get information about gene expression, you get it at the single cell, single molecule level, in fact, while maintaining the tissue morphological context. And so today, I want to take you through a couple of options that we have at ACD for ways of doing this. Whether you pick an RNAscope assay, a BaseScope assay, or a miRNAscope assay would depend very much on what you're interested in interrogating. I want to start first off just by orienting us all to in situ hybridization as a technique. It's been around for quite a long time, but I think a lot of times PIs have shied away from it because historically it's been very difficult. And then I'll talk to you about RNAscope. So RNAscope is sort of the flagship technology that ACD, Advanced Cell Diagnostics, which is part of Bio-Techne, came out with. If you're interested in looking at mature microRNA or long non-coding RNA of more than 300 nucleotides in length, this would be the assay for you. Shorter than 300 nucleotide sequences, you're looking at a BaseScope assay. miRNAscope is our most recently released approach where you can now, in fact, use miRNAscope to look at mature microRNA down to 17 nucleotides in length, 16 potentially, depending on the sequence. If you want both the transcript and the protein level information, we have an update to the ability that you have to run an ISH coupled with an immunostain, whether it's an IHC or an IF immunofluorescence if you're looking at fluorescent RNAscope. So lots to talk about, and I do plan to leave time at the end for Q&A. But just to remind everyone of what in situ actually is, you are using a probe, so an oligonucleotide sequence complementary to a sequence of interest in your tissue. The assay has been around for a long time. This is an image from a publication from 1969, where they were using radio-labeled probes to detect ribosomal RNA. Now, a couple of disadvantages with in situ. One, this used to take a very, very long time. In fact, the image here that's shown, it took them 2 to 3 months to develop these images. The other problem, of course, is that RNA is an unstable molecule as compared with protein and DNA. So unless the tissue is fixed very thoroughly or frozen very quickly, the RNA in your sample may be too degraded to detect. And traditionally, the probe that was used to hybridize to the sequence of interest was quite long, 500 nucleotides, 800 nucleotides in length, meaning that if any portion of the RNA target of interest was degraded or fragmented, or there was an RNA binding protein in the way, you wouldn't be able to see any signal. And lastly, there was a ton of optimization required because depending on the length of probe and the nucleotide composition, you would have to optimize for length of time you let the probe sit on your sample, temperature. ACD came along and basically developed an in situ hybridization assay that improved specificity and sensitivity of the in situ process. The overall workflow is very similar to traditional in situ, where the first step is to permeabilize your sample. Now, RNAscope works whether you have fresh frozen tissue, fixed frozen tissue, FFPE, formalin fixed paraffin embedded samples, cultured cells, organoids, what have you. But what you need to do to the sample to allow for probes to get access to the RNA will change depending on how the sample is prepared. After you permeabilize your sample, the probe goes on, and in a 2-hour-long hybridization step at 40 degrees, will go and find its target hybridizing to it. Afterwards, in a series of amplification steps, we build a sort of amplification tree structure on top of the oligos that make up the probe. It's not like a PCR, there's no amplification of the material. Rather, we are amplifying the signal from the probe hybridizing to your target. At the end of the day, because this is such a sensitive assay, you can see individual RNA molecules seen as punctate dots, which can then be quantified with either software like Halo, which has a fairly expensive license, or free open source software like Cell Profiler from the Broad, QPath, ImageJ. There are probably more softwares out there every day. So taking a deeper dive into the RNAscope probe design then, we are still designing a probe against a rather long portion of sequence, usually about 1,000 nucleotides. However, the probe is a pool of these Z-shaped oligos arranged in pairs. So each Z has a partner Z. The portion of the probe that actually hybridizes to the RNA of interest is quite short, at only about 25 bases in length. And when one Z binds exactly adjacent to its partner, together they form this upper portion here, this pre-amplifier binding site, 28 base pairs in length. And this, if you ever hear us talk about probe channels, it's a little confusing because you usually think of channel and you think of fluorescent channel. However, no RNAscope probe is ever directly conjugated to a fluorophore or a chromogen. Rather, when we say channel, it's just referring to the tail sequence up top here of each of the ZZ pairs in a particular probe. And because you have this sort of modular probe design, even if some of the transcript is degraded or fragmented, as long as you get a certain number of these ZZ pairs to hybridize to the transcript, you have 20 available to you in a standard RNAscope probe, but a good rule of thumb is you only need about 6 of the 20 to hybridize to see signal. So it's sensitive in that way where you have all this redundancy built into the probe design. The other place the sensitivity comes from is this amplification tree structure. So onto each ZZ pair, you hybridize a pre-amplifier molecule, which is sort of the trunk of your tree, so to speak. Onto that, then you can apply amplifiers, which will hybridize to the pre-amplifier molecule, creating this scaffold onto which we can apply so much label probe, whether a chromogen or a fluorophore, that you can see these individual molecules. Again, you have ZZ pairs tiling all along the length of the sequence of interest, each of which gets an amplification tree. There's a lot of information available on the website. It's just sometimes hard to find. You can search for a particular gene of interest and see if we have a probe for it. You can also search by a cat number if you know it. And then if you press this view details button here, it drops down more information about the probe, including the base pair start and stop, of the accession number sequence. This is actually a hot link. You can click on this. It takes you to the NCBI database, and you can see which portion of this transcript a particular probe, in this case, human PPIB probe, will be targeting. And then, as I mentioned, no probes are conjugated directly to any particular label. It's the choice of the RNAscope kit that determines how your probe is detected. If you're looking at just one-ish target per slide, you can do singleplex brown with the DAB chromogen or singleplex red with the fast red chromogen. Duplex RNAscope is available if you're wanting to look through a bright field microscope at two-ish targets on the same slide. Multiplex fluorescent RNAscope, you can do up to four-ish targets per slide, plus DAPI with this assay. And if you have access to an autostainer, either a LicaBond RX or a Ventana Ultra, you can run singleplex RNAscope, duplex RNAscope, and multiplex fluorescent RNAscope. I have High-Plex over here because High-Plex is perfect for fresh frozen or fixed frozen samples. We do not recommend it for FFPE tissues, however, and it's a manual only assay, but this can do up to 12 targets on the same slide. So if you're interested in validating single cell RNA-seq data sets, for instance, this might really come in handy. And this is what typical RNAscope signal looks like under the microscope. This is fluorescent multiplex RNAscope data, two probes as an example. One labeled in green fluorophore is a lower expressor. The other is labeled with red fluorophore. Now, again, these punctate dots are the collection of ZZ pairs and their amplification system that attach to any particular transcript. So variation in dot size or intensity doesn't tell you anything biologically relevant or interesting. So once you threshold for background, a dot is a dot. For quantification purposes, you're counting number of dots. Now, for a highly expressed target like this, where the transcript is abundant enough that the dots sort of start to pile up on top of each other and cluster, this is expected signal readout for a high expressor. And RNAscope comes in handy in a lot of different applications. I mean, there's a lot of non-coding RNA that perhaps you wouldn't have an antibody for. But the other great advantage of spatial transcriptomics is you get this information about subcellularly where are these things being expressed. And so here's just two examples where you have Gas5, which has signal both cytoplasmic and nuclear. This is singleplex chromogenic brown RNAscope. Gas5 on the left, Mallet1 on the right. This is a pretty well-established biomarker for early stage non-small cell lung carcinoma. But as you can see, it gives this very intense signal that's restricted to the nucleus. Another example, antibodies are great. If you have an antibody that you know works very well, you can put on your primary antibody overnight, the next day come in with your secondary. If you don't have an antibody, though, you can use a probe instead. And for GPCRs, for instance, here we're seeing LGR5. It's just difficult to find a reliable antibody against these targets. However, we can design a probe instead, and it takes just about 2 weeks to get a new RNAscope probe designed and synthesized and sent to lab. And for cell type identification, it might require more than just one target per slide if you have a more complex gene signature. This is a paper that just came out last year. A group at Hopkins was developing a multiplex fluorescent RNAscope assay to detect malignant prostate cancer cells in urine samples. So a lot of times the data that you see with RNAscope is in intact tissue, and here we're actually looking at a cytology sample. And not only did they use RNAscope probes for 3 markers as shown here, NKX31, PRAC1, and PCA3, but they also used an antibody stain for PSMA. So you can do ISH and protein staining together in the same sample. They also use Cell Profiler to develop an automated spot counting pipeline. Here you can see quantification data for NKX3 on the left, PRAC1 on the right in PSMA negative versus PSMA positive cells in their assay. And they did show that in patient samples, they could identify the presence of prostate cells in urine for the majority of patients, like 73% of patients. Now, compared to the 36% sensitivity with immunofluorescence alone, this is a massive improvement. Now, 73% is still lower than the use of qPCR for identifying this cell type, but it could be that perhaps incorporation of other cancer-specific targets in addition to PCA3 could increase the detection sensitivity of the assay. BaseScope is not a more sensitive version of RNAscope. However, it's an assay that can be used for certain applications. It's the same approach as far as designing a ZZ pair probe detected with an amplification system, but BaseScope probes are shorter at only 1, 2, or 3 ZZ pairs, depending on the target. And whereas with RNAscope, you have the full complement of ZZ pairs that attach to a transcript that forms a dot, each individual ZZ pair with a BaseScope assay is detectable as a single dot. It just requires more amplification steps in order to see each ZZ pair. So because we can design now a shorter probe, that opens up a lot more things that you can interrogate, including splice variants. I'll talk about that in just a sec. Short or very highly homologous sequences. If you have a gene family where there's really not a lot of sequence difference between particular family members, we will probably still be able to design a BaseScope probe for you. Point mutations, we have a list of validated point mutation probes, including KRAS or the BRAF mutations. And then a lot of folks in industry that are using gene editing approaches and in academia as well might be using BaseScope to see the results of their gene editing approach. Overall, the assay workflow is very much the same as RNAscope, where you permeabilize the sample, hybridize your probe, and amplify your signal. Again, there's more amplification steps with this assay. It's a slightly longer protocol than RNAscope, but you still see punctate dots under the microscope at the end of the day. And it still works in all kinds of different sample types: fresh frozen, fixed frozen, FFPE, cultured cells. You can look at one BaseScope target with this singleplex BaseScope kit. It's a red chromogenic assay. And singleplex BaseScope also runs on the Ligabond or the Ventana Ultra. If you have two targets you want to look at together on the same slide, you would use duplex BaseScope, which is red-green chromogenic assay. This is not automated to run on either autostainer. This is a manual-only assay for the time being. And a really good example of when you would use BaseScope instead of an RNAscope approach is if you're interested in splice variants. So sticking with that prostate cancer application, some types of prostate cancer still rely on androgen signaling, even after removal of the prostate. And androgens exert their effect through the androgen receptor. ARV7, shown here, is a splice variant that ends up with a truncated ligand binding domain, as you can see here. ARV7 has 3 exons that are the same as the full-length transcript. Exons 1, 2, and 3 are identical, but ARV7 ends up with this truncated 4th exon, cryptic exon 3, or CE3. And it's emerged as a biomarker for resistance to certain next-gen therapies available for treating castration-resistant prostate cancer patients. So you could design an RNAscope probe of 20 ZZ pairs if you restricted those ZZ pairs to being within a particular exon. So you could design an RNAscope probe to exon 1, exon 8, which would be specific to the full-length transcript. It would be shared, exon 1 would be shared by the full-length and the splice variant, but you could design an RNAscope probe to this cryptic exon 3 over here. Or you could design a BaseScope probe to target particular exon junctions. So it's a little bit more of a specific approach to detecting this the presence of this splice variant. This slide is a little busy, so apologies on that, but the top row here is a cell line that is negative for both the full length AR transcript as well as ARV7. Here in this very tiny schematic, you can see where these probes were designed. The middle row here is a cell line LNCAP, which is positive for the full length transcript, but the field views this as negative for ARV7. And then this last row is a cell line that is positive for both. And if you sort of march along the columns, you can see the probes as they were designed and the signal readout in these cell lines. So for example, the BaseScope probe that's specific to exon junction E3, CE3 here really only shows signal here in this cell line. There is one BaseScope dot that you can see here. Interestingly, this has been validated with qPCR as well for some detection of ARV7, but again, the field considers this cell line to be negative for ARV7. And another thing that's interesting, there was a paper that was published prior to the development of the BaseScope chemistry, where they used RNAscope probes that sat just within, let's say, exon 1, for instance. And they saw this very large, very intense staining in the nucleus. And the idea is that this is actually pre-mRNA, so pre-spliced mRNA, because you don't see that type of signal with BaseScope probes that are specific to just one exon junction or another, which you know would only be present in mature mRNA transcripts. And then, as I mentioned, we can use BaseScope to look for point mutations. This is a partial list of the validated point mutation probes that we have available that you can order if you're interested in doing so. miRNAscope. So this assay came out recently enough that I don't have any publications to show you. I have ACD internally generated data, which is cool and exciting, but I look forward to being able to share what the field is actually using this to do in their own research down the line. The other thing is, this is still a patent pending assay. And so a lot of the questions about the probe design approach and the specifics of the assay, unfortunately, I don't have available to share, but I do look forward to being able to share more information as it's released by our R&D team in the future. For now, what I can tell you is that it's a different probe design strategy. We're getting away from the ZZ design of RNAscope and BaseScope probes, but it's a similar idea in that you have a target-specific binding portion of the probe and then a portion that is recognized by an amplification system. You would use miRNAscope to look for mature microRNA, silencing RNA, ASO, other very, very short sequences. It is a single-plex chromogenic assay, and this too runs on the LICA bond. Unfortunately, it's not compatible with the Ventana Ultra, but if you do have a bond or a hybridization oven at the bench, you can run miRNAscope. And similar to RNAscope and BaseScope, after your probe is hybridized, you build this amplification tree structure on top in a series of amplification steps. It is compatible with protein detection as well. You can do an IHC. And like RNAscope and BaseScope, there's very little optimization that you need to do for this assay. The only portion of the assay that you would potentially need to optimize is how much or how little you digest your tissue, depending on what it is. So for example, this is a mouse embryo cross-section that we're looking at. I believe it's E15.5. And we know that miR124 is important in the developing brain. And as you can clearly see here, you have this beautiful and specific detection of that microRNA here in the developing mouse brain. This is our universal negative control probe for miRNAscope. It's a very short scrambled sequence. So it's a real miRNAscope probe. It's just not going to be able to hybridize to any sequence in the mammalian system. And this is showing detection of a couple of different microRNAs in various human normal tissues. miR205 in breast tissue, miR122 in the liver, miR21 in bladder, and miR221 in the stomach. On the right here of this panel is that same scrambled probe, so universal negative control probe for miRNAscope in breast, liver, bladder, and finally stomach. So ACD internally has tested a pretty wide range of microRNAs at this point in tissues and cell pellets, from high expressors to moderate expressors to low expressors, and we are able to successfully detect the corresponding expression level with miRNAscope, which is it indicates that the assay is in fact highly sensitive if you can pick up something that's expressed at this level. And then here's miR126 in a variety of human cancers: colon cancer, cervical cancer, ovarian cancer, stomach cancer. And then again, this is a representative image of what the negative control probe looks like for comparison. And LET7, which may ring a faint bell to a couple of you on this call, this is 3 different miRNAscope probes for a couple of the family members of LET7, LET7A, B, I. And as you can see in this rightmost column, the lowest expression is detected here with this probe in kidney, brain, and intestine from mouse. In addition to microRNA detection, previously to miRNAscope being released, we could design an RNAscope probe against the mRNA that was the target of an ASO application. But now we can design a probe that is actually detecting the ASO itself, about a 22 nucleotide sequence typically. And it's very apparent which panel here was the vehicle and which was treated. This is in cardiac muscle, and this is quadriceps of the mouse. The negative control probe I've shown you is this scrambled sequence, and then we do have this universal positive control probe as well, RNU6, that we recommend to use to assess for RNA quality and digestion conditions of your sample. I'm running out of time, so I'm going to fly through this, but suffice to say, you can do IHC. Now, previously, the RNAscope pre-treat portion of the assay wasn't always so kind to the epitope your antibody might be trying to detect. And we've reworked, or rather worked up a workflow approach to getting your antibody to play nicely with RNAscope pre-treatment portion of the assay. Essentially what you do is you start the digestion protocol, you apply your primary after the first portion of the pre-treat protocol, do a postfix with 10% NBF. Then you continue with the RNAscope probes, the RNAscope amps. You do your secondary antibody at the very, very end, and then cover slip. And we've seen that to have better chances of you being able to detect your protein and your ISH target on the same sample. And here's just one example image of microRNAscope for miR223, as well as a protein stain for CD3. So just to sum up, a lot of different options available as far as the ACD family of assays. It's a highly specific approach to detecting gene expression in your sample. It's very sensitive based on the proprietary probe design. You can look at mature microRNA now with miRNAscope at the single cell level. BaseScope you may find more useful for detecting splice variants. And then you can do both the ISH and protein detection together. And just ahead of switching over to Sarah's talk from NanoString, just so you know, you can incorporate RNAscope into your NanoString GeoMx DSP workflow. I'm sure she'll talk more about that, but suffice to say, you can use an RNAscope probe to pick your region of interest for interrogation with the GeoMx DSP. If you don't have an antibody, or you've previously been using RNAscope and you want to continue to use that marker to pull down your ROI, RNAscope fits very nicely into that workflow. So with that, I'm happy to take any questions if there are some.
Great, thank you, Emily. Any questions for Emily? Yeah, maybe I'll just start with one. Do you have a plan for attempting to multiplex with the microRNAscope?
Good question. As far as the foreseeable future, no, unfortunately, it's a single-plex assay for the time being. Definitely as we look to add more and more capabilities to multiplex, we'll definitely let you know. But for now, it's a single-plex chromogenic assay for the time being.
And one more question on that. Is it quantitative? I mean, can you actually get expression levels from the amount of stain?
Yeah. So RNAscope is, we say RNAscope is quantitative because you can count individual dots. This is the semi-quantitative scoring approach that ACD uses to quantify RNAscope data. MicroRNAscope, as you can kind of see here, the scoring criteria is different. Rather than 0, 1, 2, 3, and 4 bins, depending on number of dots per cell, it's a 0 to 3 score. Because the dots are physically smaller, they tend to cluster up faster. So it becomes a little bit more difficult to count individual dots. So I would say this is more of a semi-quantitative assay. However, even in cases where RNAscope probes are highly expressed and cluster up, sometimes researchers will, rather than count number of dots, they're looking at total number of pixels that have signal, for instance. So you can still get quantifiable data from this, but I would hesitate to go as far as to say this is a directly quantifiable assay. I think you're in the bucket of looking at more of a relative gene expression pattern here.
Great, thanks. There's a question from Jean Schaefer. She asks whether you have any experience with ISH or combined ISH-IHC for snow RNAs.
Theoretically, that should be totally doable. You would use miRNAscope to look at snow RNAs, and that picture I showed towards the end there. Sorry, let me back up a bit. So it would be this assay, essentially. You'd be using the miRNAscope chemistry to look for your snow RNA and then doing your protein stain as well.
Okay, and then one question from Simone Anafosi. She asks, do you have a standard protocol for IHC, ISH combined?
You would want to couple your working antibody protocol to your RNAscope protocol. But with the new sort of reworked co-detection approach, yes, we have a tech note I can send you, and that outlines that workflow of pre-treat depending on your sample type, primary antibody. Whatever your antibody protocol currently is, we'd recommend to probably increase the concentration as well as the time if you're not already doing an overnight primary antibody incubation. There is an antibody diluent from ACD that we'd recommend. Then you do your RNAscope and then detect with your secondary at the end. So it's kind of a combination of using your already working IHC or IF protocol and then fitting it into the RNAscope workflow.
All right, thank you very much, Emily. There are one or two more questions in the chat if you'd like to maybe answer them. Sure. You can also cut and paste links in there if people want to get the protocols and things like that. That would be great. So I think we're going to move on to the next talk for today's RNA Technology Showcase. Emily, if you want to stop sharing. We're going to have the next talk by Dr. Sarah Warren. She's the Senior Director of Translational Sciences and Research and Development at NanoString Technologies. And she's going to tell us about revealing hidden biology with spatial high-plex transcriptional profiling on the NanoString GeoMx platform. So welcome, Sarah.
Thank you so much, Dr. Frank, and thank you all for joining the talk today. It's a pleasure to be here and to be speaking with you. And I have to say that we're very happy to have the opportunity and also to have relationships already with investigators who are part of the HIRM. So I'm not going to speak about them today, but we have active project with Dr. Ioannis Vlachos. And so if you have any follow-up questions about anything I've told you today, I'm sure he'd be happy to talk with you and tell you about a bit about what his experience has been like with the GeoMx platform. So as the title of my slide indicates, I'm going to tell you a little bit about our digital spatial profiling platform that's called GeoMx. And I'm really going to be focusing my talk today on some of the newest advances that we have with the platform, thinking about our very highest plex RNA assays right now. And I'll show you an introduction to the platform for those of you who haven't seen the technology before, and then a few case studies sprinkled throughout so you can understand what you can do with it as well. So for anyone who is not familiar with NanoString, we are located in Seattle, Washington. We have been a company for, well, since 2003, so getting close to 20 years right now. And we have two platforms that we have commercialized. The first that we did was our nCounter assay for bulk transcriptional profiling using gene expression panels up to 800 targets. It's a hybridization-free or a hybridization-based approach, enzyme-free, enzyme-free detection methods. So it's really compatible with FFPE tissue. And then the second the newer technology that we have is our digital spatial profiler that's called the GeoMx. And it takes a lot of those same great features that we had in the nCounter and applies them now to spatially resolved protein and transcription profiling from tissue samples. And so why do we spend so much time doing this? It's because at our heart, we're really a biomarker company, right? And we have had extensive experience working in development of biomarker diagnostics. And we believe very strongly that there is a lot of biology that you can learn by asking careful questions of the cells and the tissues that you're studying. This is just a slide providing a high-level overview of one collaboration we did with Merck to develop a pan-tumor gene expression signature for predicting response to pembrolizumab off the nCounter platform. And it was great. We were able to develop a signature. It's called the tumor inflammation signature, but it didn't have perfect predictive performance. We saw that there was reliably in any data set we looked at, a cohort of the tumors that were inflamed, predicted to respond, but did not respond from these tissues. And so we started thinking about why that might be, what's going on with these samples. And we realized that the fundamental limitation of these bulk profiling approaches is that you lose all of the information that's captured within the tissue architecture. So here I'm showing you a picture of two tissue samples that had very similar TIS scores by our diagnostic assay, but you can see they're totally different when you actually look at the distribution of lymphocytes within that tumor. I believe this is I believe it's a colon cancer. And you can see that the sample on the left is highly infiltrated, or is highly excluded, forgive me. But the sample on the right has a high degree of immune infiltrate, but our TIS assay wasn't able to resolve those. So we really wanted to be able to have ways that we could help you understand what is going on in these tissues. And our solution for this was the digital spatial profiler. So it is a all-in-one instrument which allows you to do imaging of the tissue, molecular profiling of the tissue simultaneously using the hybridization-based approach that makes it compatible with the clinical tissues that are frequently formalin-fixed paraffin embedded and not always of the highest quality. So this is the GeoMx workflow at a high level. What we're doing is it's very similar to an immunohistochemistry protocol where we are staining the tissue with fluorescent antibodies to generate those visual images that we use to understand what that tissue architecture is. And in parallel, DNA barcoded antibodies or RNA binding oligos that we use to generate the molecular data. After we've stained the tissue, we put it on the instrument, we do a whole tissue slide scan, and then we select regions of interest for deeper molecular profiling. And within each one of those regions of interest, we shine UV light that cleaves those DNA barcodes off of the antibodies or off of the oligos, aspirate them up, collect and then quantify those barcodes on either an nCounter system or an Illumina sequencer. And so what you get at the end of the day is molecular data about the precise protein or transcriptional components of one particular region from that tissue that you were characterizing, and the corresponding visual images so that you can see with 4-color fluorescence what are the cells that are present in that region. So the whole secret behind the GeoMx system is the digital micromirror device we have, which is the element of the system that is controlling the path of the light. It's the optics, it's the heart of that optics system. And it's about the size of a postage stamp. It's 1 million independently articulating mirrors that can be custom configured based upon computationally generated tissue maps to precisely shine that UV light to only the regions of the tissue that you want to profile. So what I'm showing you here is an example of a TMA core where we have stained it with pan-CK to illuminate the tumor cells, and CD45, which is the pink stain, to illuminate lymphocytes. And we took that pan-CK yellow staining pattern and we automatically generated a mask from it. And that's what you can see at the top right of the slide here, where we've got the tumor cells are in white, everything else in the tissue is in black. We feed that mask back into the system automatically. UV light shines and it's only going to illuminate those tumor cells. It's not going to illuminate everything else. So with the same approach, you can optically dissect that tissue section from your slide without ever having to touch it. And then you go back in, you invert that mask, as you see on the bottom of the slide, and you repeat the exposure to UV light. And now you've collected all of the transcriptional or protein profiling from the lymphocytes and the stroma that are in your tissue, not from the tumor cells themselves. And so what happens is that when you start to look at that molecular data, it is very obvious that these cells, of course, have very different transcriptional or protein profiles, right? So you can see here on just a really simple heat map, right, how extremely different these tissues are. And you would never be able to see this if you were doing transcriptional profiling, of course. And if you were trying to approach it with single cell profiling, you would understand that there's a lot of different cell types here, but you'd have no idea where in the tissue they're located. So we've really tried to leverage the power of that optical mirrors and the digital micromirror device by enabling the system to have a lot of flexibility with the shapes that it is drawing within those ROIs to do the compartmentalization that allows you to segregate out those regions of the tissue. So here I'm showing on the slide just a couple different examples. I'm not going to go through them in great detail, but I'd say that the ones that are most common for us to use are either the geometric compartment profiling, which is the far left, which is just where we're collecting the entire signal that's present within that region of interest, or the segmentation approach, which is the one I just described to you, where we use one of those 4 fluorescent markers to make a mask and then use that to separate out the tumor cells. Rare cell profiling is also something you can do if you want to use a particular marker for a cell type of interest to understand, for example, what are the CD8 T cells doing in this particular area of the tumor. So it's great to be able to profile the tissues in a lot of really flexible ways, but if you don't have the molecular content at the back end, it's not going to generate much useful data for you. And so we've really spent a lot of time trying to build out the panels and the assays that are compatible with the GeoMx system so that we can give you, the investigators, as much flexibility as possible. So what I'm showing you here first is the first series of panels that we released for the assay, which were protein panels for the nCounter readout. And you can see here that to date we've built up, about 300 targets where we have done full validation on these antibodies, put them through a 5-stage screening process in our lab so that we know when we give them to you, they are going to work right off the bat. And they are organized into cores and modules that you can mix and match so that you get the content that is most customized for your particular research question. But that wasn't quite enough for everybody. And we always had people that said, but I want all the content, I want all the proteins, of course. We weren't able to do that with the nCounter system, but we were able to transfer that protein content over and change the readout at the end of the day to be able to do protein profiling as well now with the NGS readout. So really, we have now unlocked the doors for what we can do in terms of protein profiling, right? We can mix and match as many different proteins as we want. All the readouts come out the same. There's no interference. There's no cross hybridization that we see. It's a really, really flexible way to be able to measure the proteomes from your tissue. But we are an RNA company at heart. We really want to be able to leverage the very, very highly multiplexed capabilities that you can get by profiling RNAs. We love proteins, of course, but we're limited there because you have to have high quality antibodies available. And we know that's not always the case for some of the targets that are most biologically interesting. And so, in addition to all of the protein profiling we've done, we've also built RNA panels for the platform, just as I told you. And what we're really excited about now is these RNA panels that we read out with the next-gen sequencers Our first next-gen panel was called the Cancer Transcriptome Atlas. It's about 1,800 panels, 1,800 genes, really focused on tumor microenvironment profiling. Has the ability to be customized with some additional content. It has some of our transcriptional signatures embedded with it. It's a great panel if you're doing cancer biology in any way. One of the things that we have done with this panel is really to be able to develop the capability of doing cell type profiling based on the transcriptional patterns within a tissue. And so I wanted to show you just one set of data from some internal data sets that we generated, where we were looking at a lung cancer tissue section, characterizing it with the CTA panel, and then using different transcriptional signatures based on single-cell RNA-seq database data that had been published to understand where within this tumor tissue are we seeing variations in the immune cell population. And so you can see we took a gridded approach to profiling with this particular tissue. We applied our spatial decon algorithms, and we're able to characterize 18 different immune cell populations here. And what you can see is that there is a diversity and a distribution of these cells. So we have a high abundance of T cell populations over here on the left side of this tissue, whereas we tend to see more of the macrophage elements over here on the right side of the tissue. And there's no way that you could use, or I guess you could use IHC or really complex multiplexed visualization strategies to be able to resolve all 18 immune cell populations from a single tissue slide, but it would be very challenging. And this is an assay that we've done in a single day with a single tissue slide, and we got information about 1,000 other genes at the same time. So not only can we tell what are these cells that are here, but we can also say what are they doing and who are they interacting with. This is just another example where we've kind of visualized that in a different way, and you can see more clearly now where those different immune cell populations are distributed within the tissue. That wasn't enough for us, of course, right? 1,800 targets is great, but as soon as we released that, people came back out and said, I want to do more. I care about things beyond cancer. We said, that's great. How can we do this? We need to just, stop playing around and go for the whole transcriptome here. And so that's what we've done. We are going to be releasing our human whole transcriptome assay later this quarter. We will be following it up shortly with a mouse whole transcriptome atlas, where now we can look at 18,000 genes in the human tissue samples, or 22,000 genes in a mouse, single tissue sample, understand everything that's going on in that whole transcriptome. It's not 3' biased, that's really important. So where a lot of other technologies would be introducing artifact through the amplification process, we're not doing that because it's direct hybridization. We, again, like you'd think if you put 18,000 genes in a single assay, why would anyone need to customize it? But we've seen already that of course you need to customize it. So we've got the ability to customize, put in your own particular genes, your probes, your, and customization is easy, right? You tell us what targets you want to characterize, and we make the probes for you, and then you spike it in alongside the panel, and it's just a piece of cake. It's standard sequencing technology that's built off the Illumina platform, right? We've got a really close partnership with them to be able to make streamlined workflows and processes for you, so that it is no extensive learning processes as you're getting the assays up and running in your lab. It's also important to note that these panels and these assays are compatible not just with the FFPE tissue, but fixed tissues, fixed frozen tissues, any other sample type that you may have access to that you'd want to be profiling. So I wanted to give you just one quick little case study from one of our investigators at the Broad who had early access to the whole transcriptome panel. And he was really interested, this is Will Wong. He's a fantastic young investigator who's really interested in understanding the transcriptional profiles of pancreatic cancer, and especially the interactions between tumor cells and fibroblasts. And so he really wanted to understand within these two cell types, what's happening as the immune cells start to come in. So this is a story that we did at AGBT last year. It's in process for publication, so I don't have a whole lot of data to share at the moment, but what we're able to do, I just wanted to give you this stunning visual picture where we're looking at different ROIs within the tissue. And so this is one tissue section here. We have placed 4 different ROIs that you can see as each of the big bright circles. And we have profiled the cells that are within that ROI with the whole transcriptome assay. And then we're looking at transcriptional patterns within these cells based upon pathway level analysis, and looking at difference between those cancer-associated fibroblasts, epithelial cells, and immune cells. And really that heat map right in the middle of the slide, you can start to see how there are enrichment of particular biological activities within different regions of the tissue, even though they're located in very close proximity. And there's really no other way to get this level of information with the spatially resolved value-added data and all of the molecular data that you're getting other than using the GeoMx platform. So this assay is not available today. It will be out very soon. We are running it now through our technology access program here in Seattle. So it's even easier for you, right? You send your samples to our lab, they process it, they generate the data for you, and they help you then understand what it means so that hopefully you get excited about the data and want to start using it on a regular basis. But we're here, since I had the opportunity to share this platform with Emily, I did want to spend a little bit of time talking about how NanoString and ACD have formed such a close partnership to be able to leverage those RNAscope probes to really empower the GeoMx users with that technology, so that now they're not even dependent upon antibodies for visualization anymore. So we have this close partnership. We have the ability now to apply any of the RNAscope probes to a GeoMx workflow that we have. So you can use any of those targets to characterize the tissue sample you want, and then use those RNAscope probes to guide your ROI selection. I'm showing you here just a brief list of the targets that we validated, some of the kinds of questions that we're asking with it. It's compatible with the automated staining systems that you'll have in your lab already. And I don't know what to tell you, it works really well. We're using it regularly. Here's an example of lung cancer where we have visualized the tissue with PD-L1.
Yeah, this is Gordon speaking.
Hey, Gordon. Oh, sorry. So we visualized the tissue with PD-L1, RNAscope probe, PD-1 as well, CD3, and then our DNA dye. And for any of you who've worked with PD-L1 antibodies, you know that they are not ideal, right? They tend to have very poor staining characteristics. And so it's very hard to visualize tissue with PD-L1 antibody, but it works really well with the RNAscope probes. And you can see here where we're comparing just two regions of the tissue, the PD-L1 high in the middle, the PD-L1 low in the middle, PD-L1 high at the bottom, and then contrasting that to a PD-1 high stroma over on the right side of the slide. And then when we look at actually what that data looks like, comparing these two tissue sections that you see that were selected based on RNAscope profiling, but then visualized with the CTA assay, we're seeing already, very profound differences here. All of these genes that are differentially regulated in synchrony with the PD-L1, a lot of interferon signaling. And so you can imagine just taking these data sets that are phenomenally rich and comparing them on an ROI by ROI level, but then on a tissue by tissue, patient by patient level, you very quickly are able to build complex, beautiful data sets to really allow you to interrogate very deeply the fundamental biology of the particular problem that you are investigating. And then I wanted to kind of wind the talk down with just one example that we're really excited about because it is so timely for today and for here where we are here. We had the opportunity over the summer to work with Dr. Ting from Mass General Hospital, who was on the front lines of collecting patient autopsy samples from those people who were the early victims of the COVID pandemic. And he and especially the pathologist, Dr. Stone, were among the first going in there, collecting these samples and looking to understand what is going on in these tissues before we had any idea what was happening. So this is before we knew that there were coagulopathies, before we knew that there were multi-organ systemic issues, right? We just had, had people that were dying, and we needed to understand really how is the body responding to the viral infection. So they were able to get their hands on the tissue, but it was all FFPE. It had to be, right? Otherwise it was too infectious for anyone to work with safely. And there were no other High-Plex spatial profiling approaches that would allow them to interrogate the tissues. So we were able to form this partnership, characterize some of the lung tissue samples from the patients that they had. We used RNAscope probes to visualize the virus because ACD folks were the first ones to generate visualization reagents for the virus. And so what you can see here is the RNAscope generated image on the left side of the slide, where we're able to look at the pink SARS-CoV-2 regions of the lung tissue, and the GeoMx image on the right side, where we're imaging the tissue with pancytokeratin, CD45, CD68, and using visual alignment to select ROIs based upon the RNAscope probe for further molecular characterization using the GeoMx technology. And so what you can see then on the right side of the slide is actually representations of the tissue samples from 5 different patients. There are 6 tissues there because we had one case where we took 2 samples from the same patient. But it is patient, each of these is an outline of the tissue, and we are looking now at the distribution of the immune cell population within that tissue and correlating that to the SARS, the viral status of that particular ROI as determined by RNAscope. And so what you can see here is, first of all, there's a spectrum of infectivity within these tissues, which nobody's surprised at now, but was completely novel when we were doing this at the initial time. And there were some patients, who had died of virus who had very little detectable virus actually. But what we're seeing here is that there's a really profound distribution of the immune cell populations within these different patient samples. And especially if you look down here at the T cell population, that's something like this patient second from the left here, where there's absolute, first of all, segregation of the viral positive and the viral negative regions, and then profound differences. The size of the circle here is representing the abundance of that immune cell population. So for some of these T cells, for that particular patient, maybe there's about the same amount of T cell present, suggesting that there's a systemic immune response happening within the lung. But if you come over to something like the patient right next to it, where the red dots are much smaller than the blue dots, that's suggesting localized immune response. And in this case, actually, localized immune response away from the virus. And if you compare that to something like the same patient, but you're starting to look now at the monocyte macrophage populations, they've got a lot more of that macrophage response happening there where that virus is being detected. And so we're seeing an ongoing dynamic immune response happening in these patient samples as they're battling the virus and then ultimately succumbing to it, unfortunately. So you can really understand just by looking at the cell types what's happening here. And then if you layer onto that all of the other molecular data here, now we understand not just that there are T cells present, but what are these cells doing? What are the transcriptional patterns that have been activated? And we really see some really strong clustering of the tissue samples, differential expression of genes associated with immune response, but also evidence of transcriptional changes in the lung tissue itself, right? So of course the virus is reprogramming those lung tissues to become, a little more fibrotic, a little more proliferative as they try to recover from that damage. In parallel, we were able to confirm a lot of those findings because we did protein profiling on the serial section, and we saw a lot of those same targets come up again at protein, so it was sort of a self-validating experiment. So I'm going to wrap things up right now because we're almost out of time. If this sounds exciting for you, I'd encourage you to go out and look in the literature. We've got more than 30 publications that have come out so far with GeoMx technology in them, including a couple covers out there, some space in neuro, some infectious disease papers. All of these are listed on our website and you can go get the information there. If you want to know more about the platform, I really encourage you to go find our 2020 Nature Biomedicine paper where we tell you all about how the platform works and what it can do. And so I will leave you with this slide then. This is a really powerful technology. We are continually finding new ways to apply it, continually excited to see how people are incorporating it into their research. And with that, I'm happy to answer any questions.
Great. Well, thank you very much, Sarah. That was fantastic. Questions for Sarah? You can ask them in the chat or unmute yourself. I'll start with the first question that's already in the chat. This is from Elizabeth Shank. She asks whether GeoMx has been used with bacterial samples as well as eukaryotic samples.
That's a really good question. I know we've had some ongoing research with that, but I haven't seen any of the data come out. It is compatible, right? If you can get a protein or an RNA target with it, and especially if you can make your tissue work with like an IHC or an ISH staining protocol, it's going to work on GeoMx. So, you know, if that's something of interest, let's follow up online and have a discussion about what you want to do, and we'll find a way to get it done.
She says, thanks. So yeah, maybe before you leave, you could just put your email into the chat. And then just so everybody knows, Eric from NanoString did put the link for the pancreatic cancer preprint in there into the chat as well. So you can go and see what was done with the experiment that Sarah was talking about in pancreatic cancer. Any other questions? All right. Well, this has been really an amazing session. So I'm going to leave the meeting open so people can go and download some of the articles that have already been published there. Otherwise, we'll see everybody at our next event. So it'll be next week. Please, or the week after. Please join us. And thanks everybody. Thanks to our speakers, Emily and Sarah, for great talks. And their emails are now in the chat, so you can contact them for more information. All right. Bye, everybody. Appreciate it.
Thanks, everyone. Take care.